Hello everyone, welcome back to my YouTube channel. Now we will start with second session, that is female gametophyte of flowering plants. In the last session we have seen male gametophyte, the process of microsporogenesis, formation of pollen grain, etc. In this session we will see gynoecium, that is last whorl of the flower. Let us start. The unit of gynoecium are called pistil or carpel. Let us recall once. Unit of androecium, that is male gametophyte, is stamen, and unit of female gametophyte, that is gynoecium, is pistil or carpel. If a flower is having one carpel, then it is called monocarpellary. If it is more than one, then it is multicarpellary. If single carpel is present, then it is monocarpellary. If more than one is present, then it is multicarpellary. In multicarpellary condition, there are two conditions. If they are fused, it is called syncarpous. If it is free, then they are called epicarpous. I hope it's clear. Now we will see what are the parts present on pistil. If we go through the part of pistil, there is a landing portion of pollen grain that is called stigma. This is stigma. The pollen grain which are formed on the stamen, that is anther, fall on the stigma. Next to stigma, there is a long slender tube-like structure beneath the stigma that is called style. And lastly, the basal bulged part where the female gametes are present, it is called ovary. I hope it's clear, which is having ovules inside. These are ovules. We'll see in detail in the next coming session. We will see how it is shown in NCERT 1. This is a structure shown in NCERT. This is a structure of pistil, stigma, style and ovary, which is attached to thalamus. Dissected flower of hibiscus shown, pistil, other floral parts are removed. Next, this is a example of syncarpous pistil of papaver and multicarpellary, that is apocarpous gynoecium in Michelia. And this is a diagram shown in NCERT of ovule. This we will see in the coming session. Next, inside the ovary is the ovarian cavity. Inside the ovary there is a ovarian cavity. Let me draw here so that it will be clear for you. I will draw this portion here. It will be very clear. This is a bulged portion that is called ovary, which is having ovarian cavity. This all which is present here is the ovarian cavity. I am putting dots so that it's easily understanding. Hence it is called ovarian cavity, or it is also called as locule. Inside ovarian cavity there is placenta. For example, I am drawing placenta here. We have seen placentation in the PUC first year. This is placenta. Inside ovarian cavity is placenta. With arising from this placenta are these are called megasporangia. They will be arising like this. These are collectively called as megasporangia. Megasporangia. That is female gametophyte. Female gametophyte. I hope it's clear. Example, there are one ovule present in wheat, paddy and mango. Many ovules are present in papaya, watermelon and orchid. I hope this diagram is clear for everyone. Next we will see what is the structure of ovule. This structure will be explained in the coming session now. Megasporangium, it's also called as ovule. This is singular. Megasporangium is singular. This is attached to placenta. Here the placenta is present. The stalk-like structure which is attached to placenta is called funicle. And the fusion where funicle and ovule attaches is called hilum. Next, the female gametophyte are surrounded by many envelopes protective in function. Those are called outer and inner integuments. I hope it's clear. Let me repeat. The female gametophyte which is covered by protective envelopes around them, which are called as outer and inner integuments. After outer and inner integuments, there is a group of cells which is present around the female gametophyte, that is called nucellus. Next to nucellus is the embryo sac, which develop gradually from young ovule to mature ovule. At a point where integuments are absent, that is called micropylar end. Opposite to micropyle is chalazal end. I hope it's clear now. Now we will see what is the process where female gamete is formed inside the ovule. That sac is called embryo sac. After, next to nucellus, we have oval shaped structure inside the ovule, that is called embryo sac, where female gametes are present by the process called megasporogenesis. That process of formation of megaspores or ovule is called megasporogenesis. Now we will see what happens from the cell which is present in the micropylar region. This part is enlarged here. There is a large cell present in the micropylar end. The nucellus is surrounding them. That large cell is called megaspore mother cell, that is MMC. That megaspore mother cell undergo meiosis, that is meiotic one division, to form megaspore dyad. Two cells are formed at the one division. Nucellus are still surrounding, they feed the developing megaspore mother cell. Next, the meiosis 2 takes place to form megaspore tetrad, that is 4 cells are formed. We know meiosis leads to 4 cells. Among these tetrads, that is 4 cells, 3 cells degenerate and 1 cell is only present and is functional. That 1 cell will continue the process by mitotic division. By mitotic division, which forms 2 cells first. Further mitotic division forms 4 cells. Further mitotic division form 8 cells. So, mitosis first time, second time, 3 consecutive times mitotic division takes place to forms 8 cells in the embryo sac of ovule here. Next, after the formation of 8 cells, there is a systematic organization of cells in the embryo sac takes place, which is very important. Please pay attention and listen to the topic. Among these 8 cells, first 3 cells from the micropylar region arrange here to form egg apparatus. Among these, 2 are called synergids and center one is called egg cell. and other 3 from the chalazal end arrange in the same chalazal end to form antipodals. These are called antipodals. The other 2 which are remaining move towards the center. They are called polar nuclei. One of the main important feature that takes place here, the mitotic division when takes place, there is no cell wall formation of the cells. They remain free as nucleus. After the organization of cells to their end position and the polar nuclei, there is a formation of cell walls. All the egg apparatus forms the cell wall here after coming to their position. But polar nuclei do not form the cell wall. They remain as one cell, as central cell. So now this diagram is inverted and shown as given in NCERT, where the antipodals are arranged in the chalazal end. The central cell is having polar nuclei which are not cell wall formed, and the egg apparatus where 2 synergids are present and 1 egg cell at the center. The important feature of synergids is filiform apparatus which play very important role in guiding the pollen grains move towards the egg cell. Hence, egg apparatus is formed in the micropellar region. Now, this is a mature embryo sac as shown in NCERT. I hope it's clear for everyone. Any doubts, ask in the comment section. Next, here embryo sac is known as 7-celled and 8 nucleated. We will see how it is. There are 7 cells present. We'll count them. 3 antipodals, 1 center cell and 3 egg apparatus. Total 7 cells are there. 8 nucleated means 3 egg apparatus, all the nucleus are separate, cell wall is formed. Only 1 center cell, but 2 nucleus are present, since polar nuclei are not cell wall formed, and 3 antipodals with cell wall formation. So 3 plus 3 6 plus 2 is 8. 8 nucleated. I hope it's clear now. Next, now we have both microspore and megaspore, that is male gamete and female gamete. Male gamete, that is pollen grain, and female gamete, that is embryo sac. Embryo sac or egg cell we can see. Egg cell is a main cell which helps for fertilization. Embryo sac involves all the cells which support pollen grain to enter the egg cell and follow fertilization and further processes. Okay. Now the process of transfer of this pollen grain on stigma is called pollination. I'll repeat. The process of transfer of pollen grain from its anther or stamen to its compatible stigma of other flower or the same flower through any agents is called pollination. In pollination, we'll go through in detail. There are many types of pollination. Agents are required for pollination. Let me just give the highlight of pollination. We will go through detail in the next session. Pollination, there are 2 types. First is self-pollination and the second is cross-pollination. There are 2 types, self-pollination and cross-pollination. Among self-pollination, again there are 2 types, autogamy and geitonogamy. In cross-pollination, that is xenogamy. In autogamy, there are three types again: homogamy, clistogamy, and bud pollination. In cross pollination, there are two agents which are involved for the formation of zygote or fertilization. In abiotic factors, that is known as non-living things, anemophily, that is pollination brought by wind, hydrophily, pollination brought by water. In biotic factors where living organisms are involved, entomophily by insects, chiropterophily by bats, malacophily by snails, and ornithophily by birds. Now this is a just highlight of pollination. We will go through in detail in the next class. I have just introduced what is pollination, what are the types, and just go through the names of them. Okay, I will give examples for each of them and explain in the next session. I hope it's clear for everyone. Any doubts, ask in the comment section. For notes and diagrams, ask in the comment section. It will be fulfilled. And for students who need Kannada lecture, please ask in the comment section. I will post the videos in Kannada also. Thank you.